Hello students, welcome back to Trendy Achievers channel. I'm back with one more important concept in immunology, that is cell mediated immunity. Okay, so board looks very clumsy students, yes, but this is very easy. You say that the same words after the video, after my explanation. You feel that this is a very easy concept. So let's begin with the cell mediated immunity introduction. Cell mediated immunity, so cells mediating the immunity. So cells, what are the cells involved in the immunity? They are natural killer cells as well as T cells. These two cells involved in the immunity to present and process and finally to kill the antigens that present in the infected cells and cancerous cells. Okay, so let's see the process. So here the antigens are of two types, exogenous as well as endogenous. So the exogenous, okay students, before that, in the immunology there are technical terms. If you know the terms, then only the immunology become very easy. Okay, so now let me explain the related technical terms also now. So APC, antigen presenting cells, ASC, altered self cells, MHC, major histocompatibility complex 2 and then 1, TCR, T cell receptor, TH cell, T helper cell, TC cell, T cytotoxic cell, CTL, cytotoxic T lymphocytes. Okay students, there are some of the technical words which are related to our concept now. Now, exogenous antigens are processed and displayed by antigen presenting cells. అంటే ఏంటి, అవి ఫస్ట్ ఎగ్జోజనస్ యాంటీజన్ ని రికగ్నైజ్ చేసి ప్రాసెస్ చేసి ఓవర్ ద సెల్ మెంబ్రేన్ దేర్ సర్ఫేస్ పైన ప్రెజెంట్ చేస్తాయి విత్ ద హెల్ప్ ఆఫ్ మేజర్ హిస్టోకంపాటిబిలిటీ కాంప్లెక్స్ 2. Okay students, don't get confused. Antigen presenting cells process and display antigen with the help of MHC2. So that antigen is going to be recognized by T helper cells. So that T helper cells releases interleukins. So that interleukins activates TC cells. So here the endogenous antigens are processed and displayed by anti-altered self cells with the help of MHC, major histocompatibility complex 1 type. So then that going to be observed, recognized by T cytotoxic cells, and here to the interleukins, the TC cells, T cytotoxic cells releases effector memory T cells as well as cytotoxic T lymphocytes. So this effector cytotoxic T lymphocytes releases two types of proteins called as granzymes and perforins. So these perforins makes holes and pores in the affected or infected cell. So with the help of that pores, this granzymes enters into the cell and they kills antigen and finally they kills infected cell and cancerous cells also leads to apoptosis. One new term introducing you, that is apoptosis. It is nothing but the dying or killing of infected cells or cancerous cells in this process called as apoptosis. Very important term for NEET also. Okay, so now let me give you overall picture with a short. Antigen presenting cells as well as altered self cells process exogenous and endogenous antigens and processed with the help of processed and displaced with the help of MHC class 2 and 1. Then they are absorbed by T helper cells, releases interleukins. They activates TC cells to release effector cytotoxic T lymphocytes and memory T cells. So these effectors cytotoxic T lymphocyte releases granzymes as well as perforins. So they creates hole and through the pores granzymes enters into the cell and they activates enzymes called as caspases. So they kills the infected cell. Okay students, so in this way in our body they can protect the not infected cells from the infected cells. అంటే ఏంటి, ఇన్ఫెక్ట్ అయిన సెల్స్ నుంచి ఇన్ఫెక్ట్ అవ్వని సెల్స్ ని ఈ మెథడ్ ద్వారా మన ఇమ్యూన్ సిస్టమ్ కాపాడేస్తుంది. ఓకే స్టూడెంట్స్, ఐ హోప్ this is a very easy concept and little confusion to the students who don't know first. And this is a very important concept for NEET as well as IPE. Okay students, thank you.